Greetings everyone, my name is Shirval and as you are all aware of, I am the current biology faculty from Nucleon Coaching Institute, and with this video today we will be completing our series of principles and inheritance. Okay, in this we will be completing about the genetic disorders, okay, or the human genetic disorders. In that way, first I would like to draw your attention to what is a human genetic disorder, or what is a genetic disorder as to say. Okay, what is a genetic disorder? So by the name itself, okay, let me break it down for you so that it will be easier for you to understand. Disorder simply means the disarrangement, okay, or disproportion, okay, not arrangement per se but proportion definitely, okay, disproportion or disproportionate something, okay. So what is this disproportionate something? That is related to your genetics, okay. So by the previous chapters and by the previous videos you have come to know about that everything that is related to your genetics can be summed up to genes, okay. Everything that happens, everything that is heritable, everything changes that you see can be through genes, okay. Now these genes per se, okay, or these genes are contained within something of a structure called as chromosomes, okay. These are called as chromosomes, fine. These genes are contained within chromosomes, okay. So by this logic itself, okay, you can figure it out, okay, that the genetic disorder as well, okay, the genetic disorder as well is of two types. That is, one is the chromosomal disorder and the second one is gene disorder, okay. One is chromosomal disorder and the second one is gene disorder, okay. Now you may say that if genes are located in chromosome itself, why do we have to study it in two different ways? Well, how you look at a problem or how you look at something basically tells you what is your perspective. This is simply our perspective of looking into chromosomes and this is our perspective of looking into genes, okay. So continuing with this, okay, let me explain to you in a very brief sense what is a chromosomal disorder. The main gist of our topic today will be chromosomal disorders, okay. Now chromosomal disorders, okay, can be summed up or can be explained in a better way like it is a change, okay, or it is a change or a disproportionate change in that can cause something that will cause you structural change, okay, or numerical change, okay. What is this? Structural change or a numerical change? Now what is this structural change? The structural change is simply if your chromosomes get added, wherein suppose they get substituted, okay, or your chromosomes get deleted, they get inverted, or they get translocated, okay. I'll come to all of this, okay. Let me show it to you in a better way, okay. Let us talk about first structural disorder or structural changes in your chromosome. Okay, structural disorder. Now you know that disorder is simply a disproportionate means to a structural view or a morphological view of the chromosomes, and this can be explained very well in the sense that I talked about substitution. What do you understand by substitution? It simply means that during the crossing over, okay, during the crossing over, okay, during the crossing over, that means a part of this, correct? This will go to this and this will come to this, correct? This is how the exchange of chromatids or a part of those chromatids, non-sister chromatids, okay, of the homologous chromosomes occurs, okay. Now what happens is that during this change, okay, during this change, if the substitution, if the exchange happens with a chromatid segment that was not expected to come, okay, we did not expect this chromatid segment, okay, segment, not the entire chromatid. The entire chromatid can also be present, okay, it's not a big deal. But if the chromatid segment that was not expected during this crossover to come does actually come along, okay, this gives you substitution, okay. Substitution is a very simple statement, okay. It simply means that during the crossover, if the segment of a non-sister chromatid which was not expected to, suppose this is the region that was to be expected to be going under crossover. Why? Because there is the formation of your chiasmata. But what happens if this two segment, okay, does come along, okay, this is a substitution event in a structural disorder or a structural aberration. Then we move on to deletion. Although these are a bit advanced topics, you don't need to worry because these are very simple ones as well. Now do understand, something is taking your chromosomes during the crossover, or your chromosomes simply are moving on their own in this substitution itself, okay. It has been added, but if this part, if we talk about this part, what does this part go through? It goes through a lack of entire chromatid, a lack of this entire chromatid, and this is called as deletion, okay. This is called as deletion. Then we move on to inversion. Do remember, all these events are possible during crossing over, okay. Why? Because this crossing over event basically brings along two or more chromosomes together, okay, and during this crossing over event all these structural aberrations do occur, and the numerical one as well, okay. Inversion, from the name itself, inverted simply means to reverse the polarities, right? So to make you better understand, let us assume that this is your centromere, okay. This is the sequence of the gene, okay. These are all genes, okay, that are present in your chromosomes. What happens is that during the crossing over, okay, during the crossing over, this is also a site, okay, during the crossing over, this will also be the same, okay, or it can be, to make it very clear, okay, it can be this one, okay, so that it is easier for you to understand, right? What happens during this crossing over? These segments will be exchanged, okay. These segments, suppose they are to be exchanged, okay. We'll just see Okay, just see. In this, you will be getting A, B, C, D, E, no problem. Why? Because this strand is separate, okay, but this strand is going through it, okay, this strand is going through this. So, what is going to happen? That in this, A, B and C, D, E should be coming in this way, right? That this should be A, B, C, D, E. This is what a crossing over should give you as a result, correct? But what happens if this is the sequence that you get? During this exchange, the sequence gets inverted. This is called as inversion. Okay? This is called as inversion, and inversion is almost achieved, okay, or not almost achieved, it is basically happening during crossing over, okay, but not always. Why? Because there is another event, okay, that is called as translocation. What is this translocation? Translocation is also a simple fact wherein you will see that these chromosomes, okay, these have A, B, C, D, E. Let us take the example of the previous one itself. Okay. Okay. Let us take the example of the previous example. Okay. What happens is that this is the point of your crossing over. This means that this part will go there and this part should come here. Correct? What happens is that during this crossing over, if this entire part, okay, if this entire part does not come or does not arrive, okay, does not arrive at this location itself, instead it goes and binds to some other location, this is called as translocation. That means the same segment, okay, that means the same segment is now binded to a different location, not the intended point of your chiasmata. Okay, this does happen, okay, and this also leads to some sort of chromosomal change. Why? Because your gene order, the order of your genes, okay, or the sequence of your gene, okay, the sequence of your gene is called as gene order. Okay, okay, this gene order is disturbed, and whenever this gene order is disturbed, the functional protein that you are going to get from this is also changed or also modified. Okay, so what is the outcome of all these? Okay, what is the outcome of all these? This causes a genetic disorder, okay, or a structural disorder or chromosomal disorder. Okay, this causes a chromosomal disorder. Okay, this causes a chromosomal disorder. Now, in this way, we will be looking at numerical disorders as well. Now, what is this numerical disorder? There are numerical disorder of chromosomes. We are talking about chromosome. We are still under chromosomal disorders. Okay, numerical disorders can be simply, okay, first will be your monosomy. Okay, monosomy. Then will be your nullisomy. Third will be your trisomy, and this will give you the last, that is your tetrasomy. Okay, I'll explain everything. Okay, these are the 4 types of numerical disorders that you can see, that is monosomy, nullisomy, trisomy and tetrasomy. Okay, there are more. Okay, there are more. Basically what happens is numerical, we are talking about numerical disorders, right? So it basically deals with the number of chromosomes. Okay, We are simply going to discuss about the number of chromosomes. If it has increased, if it has decreased, what has actually happened. Okay. Structural can show you, or structural can give you either an increase, either a decrease, there is a change in the gene order, if there is the normal gene order, if the sequence is disrupted. Okay, all this come under your structural aberrations or structural disorders. But in numerical disorder of chromosomal disorders, you will get monosomy, nullisomy, trisomy and tetrasomy. Okay. Now, what is all this? Okay, first let us discuss about nullisomy. Okay, nullisomy simply means that, or I can show it to you like this. Okay, simply is 2n minus 2. Okay, now what is this 2n minus 2? It is basically that from your diploid complement or the diploid nature, okay, 2 chromosomes are removed. Okay, 2 chromosomes are removed. That means it can be either a entire pair is removed, okay, either an entire pair is removed, okay, and in this entire pair you get something of this nature, 2n minus 2. Okay. Similarly, in your monosomy, okay, what happens is you get something called as, or something like 2n minus 1, wherein a chromosome is simply removed. Okay. Now this chromosome is just one chromosome. It is not a chromosome pair. Okay. So if you see, what will this indicate? 2n minus 1 minus 1. This will simply indicate that 2 different chromosomes are eliminated. Okay. 2 different chromosomes are eliminated or removed, deleted. Okay. Here, however, it is a single pair that is removed. Okay, it is a single pair that is removed. That is why we have written 2. Okay, this may give you the same outcome. Okay, that is, let us suppose we talk about the human complement. Okay, if you remove 1 and 1, you will simply be getting 44 here, and you will also get 44 here. But the nature how you get 44, that is different. Why? Because here a pair is removed. Okay. However, in this case, 2 different chromosomes, like you have 1 to 22 pairs, right? You have 1 to 22 pairs of your chromosome, and the last chromosome becomes your sex chromosome, correct? This makes you 46, correct? But what happens in a monosomic condition? You simply remove out of all these 1 to 22 pairs just a single one. In a nullisomy, you will remove the entire chromosome pair. Okay, that means you will either remove the number 22 chromosome pair or number 1 chromosome pair or 2 or 3 or any of some. But in case of monosomy, you will simply remove out of those 22, you will simply remove the 1 complement, that is the 1 chromosome instead of the entire pair. Okay. Now moving on to the next one, that is trisomy. Okay, trisomy and tetrasomy. Let me explain both of these together. Why? Because if you have understood about nullisomy and monosomy, this should feel similar or familiar. Trisomy is 2n plus 1. Okay, whereas tetrasomy is 2n plus 2. Okay, do you see the similarity? There you were deleting it, now you are simply adding it. Okay, the explanation is still the same. Okay, now what is What is the purpose of me telling you all this? What is the purpose of me explaining to you all this? It will simply be that you need to understand some of the diseases. For example, thalassemia. Okay, thalassemia. What do we take from thalassemia? It is an autosomal recessive disorder. Okay, it is an autosomal recessive disorder. Okay, that is manifested or basically happens by the deletion of genes HbA1 and HbA2 on chromosome 11 and you can see it for yourself. I am not entirely sure about the chromosomes. Okay, on what exact chromosome I am forgetting about it, but we do know that this is an autosomal recessive disorder, and there are two types. Okay, you will see an alpha form and a beta form. Alpha will be the minor form, whereas the beta will be the major form. Okay, that is alpha thalassemia and beta thalassemia. Okay, now what do we take from this? It is very simple. It is an autosomal recessive disorder. That means it deals in the 22 pairs itself. Okay, not the 23rd pair, that is your sex chromosome. Okay, it does not deal with the 23rd pair, that is your sex chromosome. It deals with the 1 to 22 pair. That is why it can be, I am not exactly sure, but it is, I guess, in the chromosome 11 where you will find your HbA1 and HbA2 genes. Okay, if both of them are deleted, do remember it says that this is a recessive condition. That means if only HbA1 is deleted or HbA2 is deleted, okay, if either of these two are deleted, okay, either of these two are deleted but the other one is present, okay, then you will not get the full extent of the thalassemia, that is your beta thalassemia, but you will be getting alpha thalassemia. Okay, you will be getting alpha thalassemia. That is the concept of minor and major. Okay, what happens in this? This simply creates a faulty alpha and beta, hence the name alpha and beta globulin chains. Okay, now where do you find alpha and beta globulin chains? In your hemoglobin itself. Okay, in your hemoglobin itself. In your hemoglobin itself, okay, where you will find your alpha and beta globulin chain to be defective. What does this cause? This causes the outcome of anemia. Okay, this causes anemia, and hence you don't get, or hence you don't, and hence you need to be blood transfusion. You need blood transfusion. A thalassemia patient needs transfusion of blood if they suffer from beta, okay, at a very regular basis. Not every day, but at least once a week is very much required. Why? Because they suffer from anemia, a severe form of anemia. Okay, and anemia is a blood loss, okay, or simply faulty production of a blood. Okay, that is hemoglobin is not proper. That is why the oxygen supply is not proper. Okay, then we talked about thalassemia. The other one that we should talk about is hemophilia. Okay, although this will also cause, okay, although this will also cause you anemia, but it is a very different disorder or a very different disease, okay, than thalassemia. And the first thing that you will notice here is that this is a sex-linked, or we can simply call it X-linked recessive disorder. Okay, it is simply sex-linked or X-linked recessive disorder. Okay, thalassemia was your autosomal recessive disorder, but this is sex-linked or X-linked disorder, wherein what happens is that you have certain blood clotting factors. Okay, you have certain blood clotting factors. Okay, that is 1 to 8. You have blood clotting factors. Usual hemophilia or the most common hemophilia deals with the absence of blood clotting factor 8. Okay, and what does this do is that it does not produce the fibrinogen that will go into forming your fibrin, that is responsible for stopping or basically forming clots. Okay, and clots are required to stop blood loss. Okay, but this does not cause any clots, and hence the blood loss is not stopped. Okay, blood loss is not stopped. Okay, now let us talk about the genetics of this. Why did we say X-linked? Well, the reason behind is that this happens in the 23rd pair of your chromosomes, that is the X, X and XY. Okay, now I said that this is a recessive disease. Okay, this is a recessive disease. So that means first of all, the faulty gene or the chromosome can only be present in your X. Okay, it cannot be present on Y. Okay, it cannot be present on Y because if you see the structure of Y, okay, let me draw it for you. Y is something like this. This is your X chromosome and this is your Y chromosome. Can you see the size difference? Okay, the size difference makes it impossible for this chromosome to carry the hemophiliac gene or the hemophilia gene. Why? Because it does not have the rest of the sequence that is required for it to be containing this hemophilia gene. Okay, just a brief heads up. Okay, then moving on, if it does not have or if it does not possess the hemophiliac gene, who does possess it? That is only present on the X chromosome. Okay, but there is a catch. Therein comes the play of recessive, the word of recessive. Recessive can only be, or what do you understand by recessive is that the condition must always be homozygous. For it to express, it must always be homozygous. Can you tell me that this is a homozygous one? No. Why? Because this is not a homozygous condition, because if this was a homozygous condition, it must have this. Okay, we are not talking about this. Okay, this is a special condition that happens in the human females, which we call as carriers. Okay, they become the carriers. They are not affected by it. Okay, but they do become carriers. What happens when a carrier mates with a normal male? It's a very simple concept. Okay, this is a female, this is a male. Okay, What happens during this? We will simply show it through your Punnett square. Okay. 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 Simple. Now, what do we take from this? The thing is that a cross between a carrier female and a normal male will always give you a carrier female, okay, and a hemophiliac male. This is hemophiliac male. Why? Because the Y chromosome is so small. Y chromosome is so small that it forms a condition called as hemizygous condition. Hemizygous condition. Okay, that means Y is simply attached to a single part or a very small part of your X chromosome. Okay, and in this way, the Y chromosome is unable to give you the condition of either a homozygous or a heterozygous. So it becomes into something of an intermediate and gives you a condition of hemizygous. And in a hemizygous condition, the effective chromosome will always affect or always express whatever it has been affected with. That is why X chromosome is able to express it very clearly. Okay, this is the chromosomal aberration and the sequence. So you will get normal males and females. Okay, you will get, let me write it down for you. Okay, you will get two normal females. Do remember, why am I writing normal females? Why? Because they will not show hemophilia. Okay, they will not show hemophilia. Only one of them is affected or a carrier, but we can tell it by looking at the genotype, not by looking at the phenotype, not by morphologically seeing. Okay, you get one normal male, okay, and a hemophiliac male. Okay, you get a hemophiliac male. Fine, you get a hemophiliac male. But if you were to see it in a genotypic way, then you can simply divide it to be, you also get a carrier female. Okay, you also get a carrier female, but only when we talk about genotypic way, okay, or genotypically. Phenotypically, this is the ratio that you will be getting, 1:2:1. Okay, hope this was helpful. Okay, and the rest of the disease, that is your, I'll simply write it down so that you can go through it in brief. Okay, the rest of the disease, like your Down syndrome. Okay, Down syndrome is the trisomy of 21. Okay, what is trisomy now? You know. Okay, trisomy is simply the addition of a chromosome. Now, and since we are talking about 21, that means the 21st chromosome. Okay, the 21st chromosome will have another pair. Okay, this is your 21st plus 1. Okay, this is a trisomy of 21, and this causes your Down syndrome, which gives you moon face, cleft palate, retarded mental growth. Okay, shorter lifespan, all that. Okay, then another is your Turner syndrome. Okay, another is your Turner syndrome, that is monosomy of chromosome, monosomy of chromosome 14. Yes, monosomy of chromosome 14, which now you know monosomy means deletion. Okay, so it will be deleted. This is what you will be left with. Okay, this is what you will be left with for chromosome 14. Okay, okay, this is Turner syndrome. Then, or I can show it to you how to write this. It will simply be XO. Okay, it will be 44 plus XO. Okay, this is your Turner syndrome. So it will not be for chromosome 14, my bad. Okay, this will be chromosome for X. Okay, this will be for chromosome X itself. That means in the sex chromosome you will see your monosomy. Okay, you can simply write it like this also. Okay, in the sex chromosome, monosomy in the sex chromosome. Okay, in case of your down also, let me write it down for you. It will be 44 plus since we are dealing with trisomy and that too in your autosome, we will simply write either it is XX or it is XY, no problem in that. Okay, 2n, right, that is 44 minus, or sorry, plus 44 plus, here we will be writing about in the 21st, so 44 plus 1 plus XX. Okay, and 44 plus 1 plus xy. Okay, we can write it like this way, or we can simply write it like 44 plus xx plus 1. Okay, or 46 plus 1. You can simply write it like this also. You can simply choose to write it like this also. You can simply choose to write it like this also. All of these are correct. Why? Because they represent the correct sequence of your Down syndrome, that is trisomy, an addition. So is for your Turner syndrome. Then the last is your Klinefelter syndrome. This is trisomy of your X chromosome. Okay, trisomy of X chromosome simply will be shown like 44 plus XXY. Okay, this is the representation of your Klinefelter syndrome. So it happens in case of males, where we can see enlarged breasts. Okay, female features, reduced testes. Okay, and testosterone as well. Okay, all these are associated to your Klinefelter syndrome. Okay, there is also something called as super female. Okay, what is this super female? It is simply trisomy, but in case of females. Okay, XXX. Okay, we can simply say that this is also a super female. Okay, this is also a condition where the female, although you may think that since it is already XX and another X, so it will give her more feminine traits or more feminine characteristics. It's actually the opposite. Okay, that there is reduced chances of pregnancy, there is reduction in breast, there is a steep decline in your estrogen production, progesterone production. Okay, all these, whether it is your super female or whether it is Klinefelter, all these have reduced sexual vigor, reduced reproductive capabilities, and as such they also have less or low lifespan. Okay, all these are your genetic disorders. Hope this video helps, and by this video we come to the end of our entire web or entire series, where we started with our principles and inheritance. We talked about gene, we talked about chromosome, we talked about achievements from Mendel, we talked about the diseases and so and so. Okay, I hope this video helps, and if you have any doubts, please do let us know in the comment section, and thank you for listening.